Hello everyone, it's great to see so many of you tuning in for such an important and interesting topic today. My name is Kate Tarrant and I work for the Lower Blackwood Landcare Group. For those of you that don't know us, we're a not-for-profit independent landcare organisation with an interest in sustainable agriculture and the broader environment and river health in the Lower Blackwood catchment. You can find out more about us and the work we do on our website, lowerblackwood.com.au. Before we kick off, I'd like to acknowledge the traditional owners of the land on which we all live and work. I acknowledge the Wadandi Pibbleman people of the Bibbulmun Nation and pay our respects to elders past, present and emerging. Just a few words about how the webinar will run today. You may have noticed by now that you can hear us but not speak yourselves. That's deliberate so we can keep things moving along at a good pace. You can definitely still ask questions though, so please don't be shy, just type your question in at any time into the chat box and I'll make sure it gets asked either during the presentation or at the end. Also, if you do run into any technical difficulties, please use the status button, which is the hand you'll see on the bottom right side of your screen, to let me know and I'll see if I can fix it at our end. Don't worry though, if you do have to leave early or you drop out, we will be recording the session and I will email that out to you next week. Okay. That's enough of all the housekeeping. Let's get to the real reason you've tuned in. I am delighted to have talking to us today Mark Tupman. For over 2 decades, Mark has been active in the fields of organic and biodynamic production, permaculture, sustainability, agroecology and holistic management. And in between times, managed an orchard, animals and food gardens on his own property. Through his business, Productive Ecology, Mark provides consultation, education and training in the establishment of integrated living production systems. In addition to his consulting work, Mark works as the horticultural project manager at the Witchcliff Eco Village development and more recently as a project consultant to the Lower Blackwood LCDC. So thank you very much, Mark, and welcome, and I'll leave it to you to take it away.
Thank you, Kate. Are you going to bring the slides up? Yep, there they are. Okay, thank you, Kate. It's great to be here presenting this topic. I've been looking forward to it for a while. It's close to my heart. It's one of those things today where we've got, ever increasing prices of not just fertilizers and, all the amendments we use for fertility, but also when we have to buy in biological products as well. And then we've got, other issues in terms of how effective those products are and how effective they are in our environment as well, because, we're all in different sort of contexts and some work better than others, some are more broad spectrum. And so, there is a lot of things that we can actually do in our own situation on our farm or, in our orchards or whatever, and use these microbial cultures to improve our fertility. And, in some ways, it's not just introducing microbes, but also using them to create substances and to improve availability of nutrients. So we can use them in a wide range of sort of ways. And yeah, when we're doing it on farm, we're using microbes that are already sort of adapted to that environment. And if we do it the right way, we can actually really make quite effective and cheap cultures and use them in a way that's really going to improve, our productivity. So we've got a lot to get through, so I'm just going to dive straight into it. So why do we use microbes? They're, part of the living systems, and they carry out a whole lot of services for us, and, that really improve our bottom line and our production. And they, they're a free labor force. They do it for free. So some of the things that they do is, access and acquire, accumulate, assemble nutrients, and they put them into sort of states that are plant available, and they also cycle them so that once those nutrients are in your system, they keep on going back around again. They also carry out functions like structuring the soil, aggregation, humus production. So all of those things improve your soil in terms of infiltration and airflow, and storage capacity and that sort of stuff as well. They produce a whole range of different biochemicals that, can promote growth, regulate the way things grow. They produce compounds that suppress pathogenic microbes, and even some of the microbes that we have compete as well. And they can trigger changes in plants that help them cope with environmental stress too. So all around really beneficial parts of living systems. They're pretty much everywhere when you look around in the environment. There's microbes flying through the air, in the water, all through the soil. But obviously the greater numbers are found where there's food and habitat for those particular microbes. So classic places would be in the litter and decomposing litter on top of soil, in your topsoil, which has got quite a lot of food and, lots of habitat in terms of aggregates and different sort of, niches where they can hang out. And obviously on and around plants and animals, which are producing, plants are producing food all the time that they can consume, and animals also are eating food that they, will consume. So in the guts of an animal, they, at any one time in the soil, probably, the greater than 90% of the microbes are actually dormant. And so there's a huge amount of microbes in there, but the ones that are active are really going for it. And in a way, their activity is triggered by when the conditions are right, when there's foods available and all those sort of things. So it's up to us to create, a good habitat and, provide what microbes need. And then they will get active, they will come, and they will, sort of also express themselves in a way that's beneficial. So microbes can kind of go both ways, where, if the conditions are not right, they could actually, a beneficial microbe could turn into a pathogen and actually destroy the plant you're trying to grow, simply because it's trying to make way for something that's going to do better in that situation. However, if you get the conditions right and your plants are healthy and all that sort of stuff, it's kind of like an egg and chicken situation, and the microbes can actually work for you or against you. So really a lot of it comes down to our management of our environment. And even though today we're going to talk a lot about culturing microbes and how to go about that and use them, really the most important thing is that you actually manage your farm and your property in a way that is going to create a healthy ecosystem, which will then naturally have a range of microbes, plants and animals that work together and get to doing what life does, which is production, harvesting sunlight and energy and making proteins and carrying out all these different things that support life. In a way, what we're really aiming for is to create a farm ecology or an environment that, is sort of really healthy and carries out a lot of these processes without us needing to interfere or bring too many inputs in. And so some of the things we talk about today are kind of like ways of just sort of kickstarting that, or, supporting that whole process. And certainly, we're using products that are already there, so we're not having to bring much in to do it. But ideally, we always want to look to our management to create a healthy ecosystem that is going to support a lot of life. And some of the things that we can do, obviously, is, grow lots of different plants, which again are the energy source for all the living things, provide residues and root exudates and stuff like that. We can use a whole range of biological amendments that, are good foods for microbes, and we can practice methods of pest control and weed control that are not detrimental to the microbe populations that are there. So, using integrated pest and disease management sort of practices, and trying to sort of minimize our use of herbicides and pesticides and fungicides, obviously keeping the ground covered, trying to avoid compacting our soil. So how we manage, our traffic, also tillage, which, or aerating of the soil will have a place maybe early in a soil's sort of development, but the better your soil gets, the less you want to have to go and upset the apple cart, where you're breaking up all the layers of different microbes and the fungal hyphae that's in there and that sort of stuff as well. So obviously, going towards non-inversion or conservation tillage is something we want to work towards. Rotational grazing, which is really important, of course, because you're whatever you want to call it, moving animals on and allowing plants to recover and roots to get deep, and allowing plant diversity, as opposed to set stocking, which is going to eat out all your good plants, stop plant development, stop root development, and basically leave your soil quite exposed a lot of the time too. And, you know, diversity is a huge thing because different plants host lots of different, different microbes. So, if you're just growing a monoculture, there's only going to be a certain population of microbes that live on that type of species. Whereas if you're growing a range of different plants, especially in, like a pasture situation, or maybe intercropping or crop rotating, all of those things help to host more microbes, including using, border plants and, maybe tree belts, any way you can get a range of good plants into your system, providing, that basically growing foods, but also hosting a huge diversity of different microbes is really important. So that stuff's all the background. It's really important to focus on that management if you are going to do this. And all of the methods that we talk about today are done in sort of, in tandem with these good management practices. You can't just introduce microbes to bare, bad soil and hope it's going to do something. They pretty much will just die. They need food and life and all that sort of stuff, habitat. So there's sort of some broad group classifications that we're going to cover today, that for the purpose of what, the recipes that we're going to make and talk about, I've used these sort of groups. The first one's the aerobic soil microbes. So these are kind of the microbes that you would find in the litter layer or the top layers of your soil, where there's a lot of organic matter and there's also a lot of oxygen. And, there's a lot of microbes that we're well familiar with, all the microbes in the soil food web, so protozoa, nematodes, bacteria, saprophytic fungi, all our nitrogen fixing bacteria, the ones that are free living, as well as, some of the ones, the rhizobia. And, these microbes are really prevalent in the topsoil, and we can sort of emulate that situation by, creating sort of compost heaps that, have a lot of organic matter and, kind of like a cake for these sort of microbes. The important bit is that it's it's got aerobic conditions, especially in the initial stage of composting. So we'll go into these groups when we talk about how to culture them in a lot more depth, but yeah, that's the first group. A lot of this aerobic sort of soil microbes is talked a lot about by Elaine Ingham in the Soil Food Web circles. Another group that's really important is the colonizers, ones that actually live on plants. You've got your rhizobia, which are well known. They fix nitrogen. They predominantly live on legumes. And you've also got your mycorrhizal fungi that, again, lives off plant roots and goes out and accumulates nutrients from far and wide. All of it, helps with soil structuring and aggregation. And yeah, I mean, the thing with these is they actually live on living plants. However, they do have dormant states that they go into when the plants are dead. So if they're growing on an annual plant and it, finishes its life cycle and you go into a dry season, they will then go into a dormant state, reproductive state, that they can come alive again when the conditions are right. Just going back to, when they're talking about these ones, obviously the decomposers, what they're doing is they're breaking down organic matter so that it's cycling the nutrients back. They call it mineralization, and making it available for the plants again. So, when you've got plant matter that's just sitting on the surface, it's not going to do much for plant growth. But once it's decomposed, those minerals that are in it become available again. And at the same time, they're also sort of breaking down organic matter. They're creating these sort of humus fractions and stuff like that, which also contribute to, soil holding capacity and structure and all that sort of stuff as well. I just missed that on that slide before. And then another group that we're going to look at is the facultative anaerobes. And these are a group of microbes that can live in situations where there is oxygen, but they also survive when there's a bit of a lack of oxygen. So, in the soil, there will be areas between, say, the soil aggregates where there is plenty of oxygen, but then inside, an aggregate or inside the rhizosheath around roots, there's often, a lot of microbes who are using all the oxygen up in it, and it can become quite oxygen deprived environments. And they have this ability to switch the way they metabolize and use other nutrients instead of oxygen to burn fuels and get the energy that they need. So it's basically a process of they're switching between aerobic respiration, but then they can switch through to anaerobic respiration or fermentation as a way of getting the energy they need out of the fuels that are in the environment. And we're quite familiar with a lot of these. We make a lot of products that use these organisms, so yogurt or sauerkraut, sourdough bread. And you also might be familiar with things like effective microorganisms, which people use to decompose waste and that sort of stuff. One thing about these organisms is that they are associated with the creation of humus. So when we're talking about the aerobic ones, which I talked about before, that are decomposing and mineralizing organic matter, a lot of those minerals are basically released back for plant growth, and a lot of the carbon is released back to the atmosphere eventually through the process of respiration, where you breathe in oxygen and release carbon dioxide. However, through the fermentation pathway or the anaerobic respiration pathway, you're actually not getting rid of all the carbon in the system, and that's where you create these stable carbon complexes in the soil. So there's just a sort of quote there from Wikipedia: although the terms humus and compost are informally used interchangeably, they are actually distinct components of different origins. Humus is created through anaerobic fermentation, while compost is the result of aerobic decomposition. So we're talking about aerobic soil microbes in one situation, and we're talking about facultative anaerobes in another situation, and they just perform these different functions, complementary functions. So we want to know how to get, a lot of these organisms active on our situation, and there's a bunch of different type of cultures that we can practice to do so. One of them is making compost, and when we're making compost, essentially we are culturing a huge range of different microbes and possibly even worms as well. You can add worms to a compost heap once it cools down. But at the very beginning, it definitely has to be an aerobic process, because a lot of the organisms that we're trying to grow in a compost need oxygen. So the saprophytic fungi and the protozoa and a lot of the bacteria, all of those things need oxygen to survive. And what happens when we make a compost heap, we pile a huge amount of organic matter together, proteins and carbohydrates and all the things that microbes need, and they come and they start consuming these rapidly. And in the process, they're using up oxygen through the respiration pathway, and basically they can actually suck the oxygen out of a compost heap and suffocate themselves. And then the compost will go anaerobic, which is not conducive for those organisms we're trying to grow. So one of the most important things when we're making compost is to keep it aerobic, especially in that initial stage when there's a lot of activity and they're using a lot of the oxygen up. So they call that the thermophilic stage, which is when the compost is really hot. I have written a blog about, or a post about, making compost, which I've just put a link to down the bottom. And, I'm not going to go in depth into making compost now, but you can certainly look at that. And another thing is that we will attach, when Kate sends out the recording, there will be a number of documents I reference during this presentation that we will make available as well. I'm just covering some of the basics. So when you're making compost, in order to keep it aerobic, you can do a couple of things. There's, again, the soil food web method that sort of Elaine covers very thoroughly. And that's, when your compost is hot, after a certain period of time of heat, you have to turn it. So based on the temperature, it might be after 1 day, it might be after 2 days, it might be after 3 days. And by turning it, you reintroduce oxygen, and you also mix up your materials that might not have been in the middle of the compost heap, get into the middle, and that helps them to break down quicker and that sort of thing. And basically this turning process carries on for as long as the temperature gets above a certain amount. So once it sort of gets down below 55 degrees, it no longer becomes critical to turn your compost. And actually at that point in time, you want to leave your compost sitting, not turning it, because when it cools down enough, fungal hyphae will start to colonize the heap. And every time you turn it, you're going to break that hyphae up. And good saprophytic fungal hyphae is one of the hallmarks of fine compost. And I've got a picture of it on there that, me and Kate actually looked at some compost, a Johnson Su compost through a microscope, and that's sort of what we're looking for. So yeah, it's sort of often people make compost the wrong way around. They kind of make it, they don't turn it in the initial stage when it's hot, and then, you know, they wait, and when it's cooled down, they sort of think, oh, I might turn my compost heat now, and they go and turn it, and by then it's already kind of gone anaerobic and fungi started to colonize it, and so all it really does is destroy the fungal hyphae and breed, you know, a higher population of anaerobic organisms that you don't really want. Another method of keeping your compost aerated is by providing air channels, and the Johnson Su is a great example of this, where you're putting, you know, pipes in the heap that you're taking out after a while that sort of create these chimneys, I guess, that air can circulate from underneath the heap up through the heap. And, you know, the placement of these chimneys is no more than a foot apart through the what's like a vertical column heap. And so that's another way of sort of having a compost heap that's aerated, but at the same time you don't have to turn it, so you're not breaking up the fungal hyphae. So it sort of it addresses both those things that are integral to quality compost. Another thing about compost is that it's really important that it's mature. When you're using it to make products, you want to have had several successions of different microbe populations go through it, because every time a microbe population exists and then finishes or runs out of food or gets overtaken by another population, basically the contents of the cells of those microbes get released, either through, you know, another organism consuming them, or through, you know, when they go into a reproductive stage and they release spores or cysts, and they, you know, their cells sort of rupture and they release all these metabolites and beneficial compounds and that into the heap. And they also release dormant, you know, reproductive materials, which are much hardier and can survive out in the environment when you introduce them. You know, say you're putting out compost and the conditions are not quite right. If the organisms are alive and active, they might not fare so well. But when you're putting them out in a dormant form, as they would be in a mature compost, where a lot of the foods have been consumed and the organisms have gone dormant, they've got a much better chance of surviving out there in the environment. And then along with that, there's all these beneficial compounds like enzymes, hormones, you know, biochemicals and all that sort of stuff that's been released through this process of this maturing compost. And those are really great biochemical signals that stimulate biology and plants when you apply it in fields. So it'll stimulate plants to start exuding, and it'll stimulate microbes to start, you know, colonizing roots and getting active and acquiring nutrients and doing all the things that they do. So compost maturity is really important. So yeah, I encourage you to learn how to make good compost. Another thing about compost is that when you're making compost with materials on your farm, you're inoculating, in a sense, your compost with microbes that are already known to do well there. So when you're using plant material that you've grown, and you could use a bit of soil in your area, manures from your area as well. And those are microbes that, you know, survive there. And so when you're making compost, you're effectively, you know, brewing up or breeding or culturing a huge amount of microbes that do really well in your environment. And, you know, especially if you're getting materials from healthy environments and adding them to your compost heap, that's another way of bringing in, you know, good microbes into your situation. You can add minerals and soils to your compost that, you know, might host different mineral solubilizing microbes and things like that. So you know, basically it's a real good sort of like culturing process by making compost, of just getting huge populations of microbes going and their byproducts, and then putting them out in a dormant form to both inoculate but also stimulate biology in your soil and around your plants and on your plants. Obviously making compost is a bit of work, so you know, you don't want to have to make compost and then spread it out heavily. So you know, some of the strategies that people are using these days is making extracts, which is basically mixing compost with water. You can add foods to it and so on. We'll go into the recipes in a sec, but you're basically using mature compost, where again, it's dormant microbes that are in it and all their chemicals, which are quite stable in a liquid medium. If they're living aerobic microbes and you put them into water, they suffocate. So you really have got to use mature compost. And it's the same with vermicastings. A lot of the time, the juice around a worm farm is just rotting vegetable juice. It's not great, and it's full of dead microbes that have suffocated. So you really want to take your worm farm through to the point where it's fully mature worm castings before you make a liquid brew out of it to put out. And especially if you're going to store that liquid, obviously better to put it out fresher, but if you're going to store it, it has to be made with mature. And, you know, the products that we buy, like worm juice and stuff like that, you would hope that they've been made with very mature vermicastings. So I've sort of talked a bit about a lot of that. The most effective time to use compost extracts or compost is when you're planting or in the early stages of plant growth, because you establish a healthy plant microbiome from the beginning. So, you know, microbes colonize the roots, the leaves, the stalks, they're actually inside the plant. And it just gets the whole thing off to a good, healthy start, which, you know, all living things really benefit from in life, and, you know, prevents them from succumbing to, you know, infestations and attacks from pathogens. And, you know, it just gets the whole nutrient sort of acquisition process happening, and, you know, that bridge between plants giving microbes sugars and microbes bringing back all the things that plants need in exchange. Also, there's less surface area on young plants and seeds. It's very easy to coat seeds and do all that. So it's very easy for a little bit of compost to go a long way. And in many ways, when we're applying compost, we're sort of emulating what happens in nature, where you've got these layers of litter on top of, you know, the decomposed litter or the duff layer, and then you've got topsoil, and that duff layer really, which is broken down organic matter, is similar to the product that you have when you make compost. And again, it's full of microbes, dormant microbes, and biochemical compounds. And often when a seed falls from a tree or a plant, it drops through the coarse litter and it lands up in that duff layer and germinates in that layer of duff, or, you know, what effectively is compost, and hence gets colonized from the beginning, is able to, you know, get access to a whole lot of biochemicals and enzymes and hormones and all the things that it really needs for a healthy start. So when we coat seeds or put down extracts, or even just use a bit of compost when we're planting seedlings or use it in our potting mix, we're kind of emulating that, and we're sort of stimulating the biology, both the plants and the microbes in the vicinity, to start, you know, doing what they would do in a healthy ecosystem and create a really strong plant microbiome from the beginning. So it's a good little trick. A little bit of compost put in with a seed really gets, when the seed germinates, it will start putting out exudates straight away, because it's got these biochemical signals that it would have received in that environment that makes it think, hey, there's microbes around, I've got to feed them, and then they're going to support me. And it also attracts microbes to the situation as well. You can also use compost just without plants, just on the soil and surface litter, as a means to sort of, you know, improve. Again, it stimulates microbes, but also inoculates it, so that processes like decomposition, humification, aggregation, nitrogen fixation, etc., all get sort of kick-started. So, you know, you can sort of mix a compost extract with, you know, other biostimulants like fish hydrolysate or seaweed and molasses, sometimes some humate products, and apply those to the soil. And especially in autumn, or when the moisture conditions are right, it will stimulate all these processes, these soil processes. So it's kind of like primes the soil, so that when you plant things in it, it's already got, you know, it's a lot more functional, and plants get off to a better start, and you have better water infiltration, gas exchange, and again, holding capacity of minerals and so on. So the recipes I will give, you know, will be sent out to you. So I'm not going to go into the recipes in detail today, because it's just too much information for the time we've got. But to make a compost slurry or an extract, it's pretty simple. It's really just mixing it with water. You can add minerals and clay to it as well. Make a bit of a slurry or an extract, which is a bit runnier than a slurry, and then just, you know, strain the large particles out of it. And then you can add foods to it just before you put it out. The important thing is not to add foods to an extract if you're going to store it, because the microbes, the dormant microbes, will wake up, eat the foods, start respiring, and suffocate themselves again. So you add foods just before you put it out. And then as you spray it out, of course, they're coming back into contact with oxygen, and they can actually proliferate and grow out in the environment quite happily. You can coat your seeds with, again, more of these stimulants and so on as well. So the next recipe is just an extract, which is just a runnier form of the slurry. and I've just got the rates and everything there, but we're not going to go into that now because, yeah, you can have a look at those recipes. And there is a good chance that we're going to do some practical sort of workshops on the ones that are most required from this presentation down the track. So look out for that. So then we get into the colonizers. So it's the specific ones, you know, there's things like viruses and all that colonize plants, and a lot of actually plants in general are colonized by a huge amount of microbes, just like us. There's, you know, microbes through all parts of a plant, but the ones that we particularly want to focus on today are rhizobia and mycorrhizal fungi, because they're both really big players and they're really critical. Like if a plant is growing without its a legume plant's growing without its rhizobial associates, it's really at a disadvantage. And same if a plant is growing that normally, you know, would do so in association with certain mycorrhizal species. If they're not there, it's really compromised because, you know, in the case of rhizobia, they're fixing nitrogen, enabling the plants to make protein and so on. And in the case of mycorrhizal, they're, you know, going out and getting nutrients from all through the soil. You know, they've got a lot more access, a better ability to acquire them off soil particles and share them and so on. And it just increases the network, you know, exponentially when you've got mycorrhizae involved. So I've just got a picture there of, it's like a cover crop that I grew, and you can see, I'm just having a look to see what the nodulation's like on the legumes, but this cover crop has a wide range of species in it. It has not only legumes, but also some cereals, and I've also got some other sort of forbs in there, like there might be some some linseed. There's a few brassicas and stuff in there as well, probably clovers. And basically this is just a broad spectrum, you know, like when you've got plant diversity, you've got microbe diversity. So we're actually growing a huge, we're supporting a huge range of microbe populations beneath the soil. And because these things are growing off living plants, you then have the ability to maybe, well, they're one of those things that you can't just, you can't grow in a compost heap because they require a living plant to actually grow and reproduce. So the only way to really culture the colonizers is with living plants, and preferably living plants that are known to host a wide range of different mycorrhizal fungi or rhizobia. So they only, like I say, they only survive and grow on living plants, but, well, they only grow on living plants or are active on living plants, but they can survive and spread in the dormant state. So when a plant finishes the end of the growing season in an annual plant, the mycorrhizal fungi will release spores, you know, bacteria will release cysts, and that enables it to survive when the conditions are not right. And a lot of the time, these reproductive parts are within the plant, or they're immediately in the soil immediately around the plant. So you'll find mycorrhizal spores within the plant roots and in the immediate environment just outside of the plant roots, the rhizosphere. And so if you're growing a range of different plants that are known to host mycorrhizal fungi, for instance, and that plant goes into dormancy, you can literally dig up the plant, and within the root fragments and the soil around the roots, it has a whole lot of reproductive material for mycorrhizal fungi. And if you're going into an environment which has been denuded, or, you know, just hasn't had living plants in it for a while, or it's maybe been a monoculture environment where it wouldn't have the mycorrhizal species that you're wanting to have there when you grow different crops, you can use the root fragments or the soil from a healthy environment or an area where you have grown these plants before that you know has good healthy populations, and you can literally spread the soil and the plant roots around, or mix them in your potting mix. You can mix them with compost and apply them. You could mix them with in an extract and apply them that way as well. So you can make an extract with water out of them, and then you know, probably not as good as using the actual substrate itself, but it still might be one way of getting it out further afield. Now, when we're doing this and when we're making compost, we have to realize we're doing a broad spectrum sort of culture. So we're not specifically, you know, I mean, you can grow a legume that, you know, hosts specific types of rhizobia, and you can inoculate them with specific types of commercial rhizobia, and then you know those are in the system. But generally speaking, we're growing, you know, plants that are known to host a wide range of different microbes, and also using diversity of plants so that we cover all our bases, and we've got a huge range of these microbes that we're actually growing. And then when we introduce them to a new environment, you know, it's sort of survival of the fittest, the ones that are most suitable will take, and so on. So it's kind of like it's an approach where we're culturing a wide range of microbes rather than specific microbes, but it's very effective, it's cheap, and the microbes survive because they are in substrate when we introduce them, like compost or in soil or root fragments. So they're already sort of in a kind of like habitat that enables them to survive. They're in dormant form, and then provided you provide the right conditions and you grow plants that they can live off and on and around, and organic matter in your soil and that sort of thing, when you put them out in your environment, they've got a really good chance of taking hold. And that's quite different to some of the products that you can buy, which cost a lot. There's transport costs involved, there's waste in the plastic containers and so on. And then, they might not actually perform that well anyway. So, it's such simple technology that we can all do that's really effective. It helps to kind of get to know, what species might live on certain plants. So like obviously our legumes specifically rhizobia specifically live on legume species, and there are specific rhizobia that live on specific species of legumes. But some legumes will host quite a wide range of rhizobia, and there are also certain rhizobia species that will colonize a lot of different sorts of legumes. So we could grow a range of legumes, and you'd probably have a good chance of hosting or being able to culture a lot of the rhizobia species you need by doing that. It's the same with mycorrhizal fungi. There's a lot of plants that are really well known to host endomycorrhizal fungi species, and that's the fungi that actually burrow into root cell. The hyphae go into the cells of plant roots and then out into the soil as the endomycorrhizal fungi. And some common sort of well-known host plants that host a wide range of endomycorrhizae are things like sorghum and millet, oats, so like cereals, and then things like sunflowers, for instance, are notoriously good mycorrhizal hosts. And then you've also got, your ectomycorrhizal fungi, which is more commonly associated with tree species. And that fungi doesn't actually go into the cells of roots. It sort of more goes between cells and around the sheaths of the roots and stuff like that, and then out into the soil. And, things like pines, for instance, are really well-known hosts of a wide range of ectomycorrhizal fungi. So we can literally, if we are already growing or we already have healthy environments where we know there's going to be a wide range of microbes, and we grow these plants specific to what we're trying to achieve. So if we're wanting to grow, for instance, a cereal crop in a new environment, we could use the soil or the root fragments from oats that are growing in an environment that's already healthy and established, and use that as an inoculant in the new environment. We can also specifically culture things by, you could introduce inoculants like in the form of mycorrhizal spores or rhizobia, and you could grow like a cover crop, which is a diverse mix of the species will host these organisms. Or you could even just grow them in like sacks of substrate and inoculate them. And then once those plants have finished their life cycle, you literally just take the soil and the plant fragments from the sacks or the pots or the cover crop area where you've grown them, and again just sieve them and cut them all up and add it to your spread, put it in your spreader or in your compost and get it out on the field where you're wanting to introduce these organisms to. Quite simple, really, but effective. And then finally, the facultative anaerobe culture. So these are, again, like I said, we already are dabbling in these just on the home front when we make a lot of the products like beer and sauerkraut and yogurt and all that sort of stuff. So, they're not unfamiliar organisms to us because they can live in the, with oxygen and without oxygen. They tend to be quite hardy and resilient organisms. So they're all over the show, in the environment. And yeah, pretty easy to, once you get a culture going, like, a good culture, they're pretty easy to keep happening. they can use them in a number of ways. So we can make brews, and we can use them to digest organic materials. So like you can make liquid fertilizers like sort of fish hydrolysate or seaweed or whatever. So it's basically a matter of just getting this culture going and then feeding it and adding in organic materials that you want them to break down and convert into like a liquid fertilizer. They're also really good because they release a lot of organic acids. These facultative anaerobes, I mean, all the microbes do. They release organic acids, which play a big part in converting and chelating, nutrients or minerals into nutrients and into states where they're plant available. So, some of the nutrients are only in certain states do they become readily available for plant use, and the organic acid medium, which is what you would get organic acids out in any good healthy topsoil, or where there's decomposed organic matter and microbes, and in the process, soil minerals are converted by these microbes and the organic acids they release into the right state, and they're chelated, which is into stable molecules in the right state that plants have been taking up for millennia. And in a way, when we have just our salt fertilizers and we're putting them out, they're not in that state. So if we can convert them into the right state that plants really recognize and they're plant available and they've got carbon and nitrogen molecules attached, they become molecules that are readily recognized and utilized by plants, and take less energy on behalf of the plants to convert them into the forms that they want to convert them into as well. So we can make brews and add nutrients to these brews, sulfates and salts, and then they get converted and chelated and complexed, and then we can apply them out in the field, and we can use a lot less of them because they're so plant available. They go a long way. Obviously we've talked about the fermentation of various foods and beverages using facultative anaerobes. You can also use them to break down organic matter materials. There's certain composting systems like Spice and Bokashi that do that. It's quite common in Asia and Korean natural farming. They use a lot of this culture. You can use them to remediate waste products, especially things like oil and fuels and things like that, because they're, like I say, they're a very resilient organism, and they're really good at sort of breaking things down and surviving. They produce fuel byproducts, when you use them in a digester. It can be some of them are probiotic supplements for livestock. With livestock, they have in the guts, there's generally a sort of environment that's sort of oxygen deprived. So it's a lot of the organisms in an animal's stomach are facultative anaerobes, and so some of the probiotic supplements, are full of facultative anaerobes that are beneficial for your livestock gut microbiome, and they are also supposedly can be used as a spice stimulant in fields as well. So when you apply it to plants and to the soil and stuff like that. So like I say, they occur everywhere in the environment, and we can get starter cultures from the environment. The first one, native microbe seed, is just a culture where you go out into a healthy ecosystem where there's lots of litter, especially a forest, and you find those white sort of strands of mycelium in there, and that's sort of like an indicator that there's a good populations of what we need. And then you kind of put them with foods which are normally in the form of bran and molasses and a little bit of moisture, and you shut them off in a drum airtight because they're facultative anaerobes, and they sort of brew up and colonize the substrate, and then you have that there as a starter culture. So again, you're using microbes from your environment, but just culturing them up in the right conditions with the right foods. You can also use fresh rumen contents from an animal, which again is full of facultative anaerobes, and you can also use fresh manure, although it's less reliable than the rumen contents, because manure, once it's been out in the environment, it has to be really fresh for starters. So, it's got the exact populations you need. There's another brew you can make called lab serum, which is just a brew that's cultured on starch in water, so white rice in water, and you leave it with an open lid to, like microbes, facultative anaerobes will come in from the air and colonize that water because of the starch and carbohydrates in it. And then you mix it with milk, and it separates into whey and curds, and then you take that whey out, and that's your lab serum. So it's full of lactobacillus and a whole range of other facultative anaerobes that are really good. You can then sort of add molasses to that to stabilize it. There are commercial products available too, like BAM is one that gets around in our circles. Kefir is a culture as well that you can just readily buy from the supermarket. You just want the natural kefir with nothing else in it, just milk and the culture. You can buy effective microorganisms, which are full of a wide range of these sort of facultative anaerobes. And yeast is another one that you can buy and use as a starter culture. And then basically, it's just a simple matter of using that starter culture and putting it into a vessel and adding liquids and foods for them, and then putting an airlock on there. This is just a photo of a 200-litre drum. It's got that amber orange sort of look. It smells nice and sweet when you've made one of these brews. This is sort of like a biofertilizer that's been, the recipe was initially developed by a whole mob in South America, mass humus, and they've sort of been adopted around the world now and used in various contexts and ways. But yeah, again, it's just a barrel full of water, a starter culture, and then you add the foods. And like I say, I don't want to go into recipes too much today, but just to give you a basic idea. And again, these recipes will be posted out to you so you can use them. They're quite simple. So we've added some starter culture to the barrel. We then add 4 liters of molasses, which provides, all the energy that they need, short carbohydrates. We add 8 liters of milk or whey. These figures, they can be varied. I find it's quite adaptable as to what they will survive off. But this is a recipe that I use as a starter culture that just works really well. So I sort of stick to it. The milk's got proteins in it and calcium and various things. And then you can add your baker's yeast, which is another sort of culture. And you can add extras like, various minerals and fish meal, seaweed, humic substances, ashes from bones and grass and things like that. And you can just add them to the barrel, but if you don't want them to sort of like make your barrel full of all sorts of larger particles, it's best to put them in a fine mesh sack and then take it out after a week. And you basically just put all those things in the barrel with water, top it up, and then you close it off, and you need an airlock because the microbes will start to grow, and they will release, they will be literally releasing gases as they're doing so, and these will bubble out the airlock. And in the process, it's becoming it's a very anaerobic sort of environment. It becomes very acidic and reduced, and that's the exact environment we're wanting to create to do the sort of things we want to do. When they go through all these cycles, eating the food, and ultimately end up running out of food, and then they go into reproductive sort of stages, and like same as what happens with compost, cells rupture and they release spores and cysts into the mix. So dormant reproductive sort of materials for the different microbes that are in there, and in the process of their cells rupturing, they're releasing all these metabolites, amino acids, complex nutrients, and phytohormones, vitamins, enzymes, etc. Now, because you've created this great liquid full of organic acids, it's a very useful one to mix with salt fertilizers like we talked about, and it then can be applied in the field and make for readily available nutrients. So I've just put up a really basic recipe here where you would get, for 1 hectare to apply a liquid sort of foliar spray out to a hectare, you'd get a 20-litre bucket, and you'd get 20 litres of, or up to 20 litres of biofertilizer in there. There are other useful agents that are full of organic acids like fulvic acid or fish hydrolysate that also are chelating and complexing agents and reducing agents that, again, all help to make the nutrients, get the nutrients in the right state. And as you can see, all of these products are sort of byproducts of organic processes, the fulvic acid, the fish hydrolysate, and the biofertilizer. So you basically mix up one or more of these in a bucket, and then you add the nutrients that you want to get out. You top the bucket up with water if you haven't got it full, and then add the nutrients you want to get out, and you apply, you put in as much as you'd need to put out on your hectare, and then you sort of mix it all up, and in doing so, the nutrients get converted into the right form. You might see some bubbling off of oxygen and that sort of stuff, and then they, tend to get converted and chelated or complexed, and they're in a quite stable form once that happens. And then you've basically got to get your spray tank ready, fill it up with water, adjust the pH so that it's between 5 and 5.5, which is extremely important because if you have got bicarbonates in your water, they can then react with the fertilizers that you've just mixed in there and make them plant unavailable again. So your rainwater's pH between 5 and 5.5, no bicarbonate, so you can just use rainwater as well. But if you're using bore water or dam water, and especially if you've got issues with alkalinity or bicarbonates and stuff like that, you really do need to get your pH down to between 5 and 5.5. And you do that shortly before you spray it out, because if you leave it just sitting in the tank for too long, oxygen gets back in, and it all sort of starts to neutralize again. So basically, once you've got your pH right in your spray tank, you add your 20-litre bucket with your nutrients and your biofertilizer in it. If you want to, you can add a surfactant or spread a sticker agent that helps things just stick to the foliage of the leaves. And then you spray it out early in the day or towards dusk, preferably when it's not raining. And in this form, these nutrients get taken up by the plants very quickly and very effectively. So you really are saving yourself a lot of money, because you're getting it right into the plants, and it's cheap, because you're buying the nutrients in their raw form, and you're making a brew yourself that if you bought, a 20 or a 200-litre drum of chelated trace elements or whatever, it's going to cost you a huge amount more than if you just use simple things like biofertilizer and the sulfates from the shop, and then, follow the process. So it's an excellent way of feeding your plants instantly. And you can use these foliar sprays to increase photosynthesis, increase really sort of fast track your protein synthesis, and it can be that effective that you can have instant responses. You can put out a foliar spray, and within 24 hours you can have better higher Brix readings in your plants, which is showing that the photosynthesis has gone up. You can have good protein conversion, and you might have certain pests that can't metabolize proteins that will just disappear overnight when you get your sprays right. So normally the foliar spray program is done in conjunction with some sort of plant testing, like plant sap analysis or something like that, which helps what to put out. But there are also just broad range trace element mixes you could put out, or broad a mix that is just going to have the nutrients that are essential for photosynthesis or protein synthesis or something like that. So yeah, really handy way of going about things really cheaply. So like I say, all of these are quite broad spectrum microbe cultures, and you want to kind of hedge your bets and implement a wide range of practices and cultures on your farm that mean you've got huge amount of diversity on there, and all the things that you need to support that diversity. And then you kind of let the balancing act take care of itself. Your environment will obviously be more suitable to certain populations. There'll be plants that grow better in your environment that associate with certain microbe populations, and so on. And so you're just really putting in good management practices, and then you're using some really cost-effective tools to kind of boost the biological activity, and in some cases introduce biology to the situation if it's not there because your landscape has been degraded. One of the most important things, of course, is to grow a diversity of plants, because again, they host different microbes. And if you want to have lots of different sorts of rhizobia and mycorrhizal fungi and various different microbe groups that live on certain species of plants, you want to get those into the system if you can. Obviously, in a pasture situation, the diversity is great for animal health, but also for microbe health and function of soil. And if you are growing crops, you might want to think about integrating cover crops in between seasons or interplanting. You might also want to think about, again, things like tree belts, and in orchards, you want to mix up systems, ground covers, pioneer plants, etc. Just any way you can. If you've got livestock like chickens, you kind of want to have plants growing around the chook pen, in the chook pen, separate areas so the plants can grow before you put the animals in there and get them in and out. And then you've got diversity of animals as well, which also are hosts to a wide range of microbes. So yeah, diversity is really important. and any way you can integrate that into your production enterprise, is good. And then basically, once you've got a diversity of plant material, you've also got a huge amount of material to use for mulch and compost, which, again, is we've talked about the benefits of doing so. And then you've also got from that plant material, when the plants are dormant or die, especially the annuals or even living plants, you can go out and get root fragments and soil from the rhizosphere of mature plants to inoculate, your growing environment. And once you've got a whole bunch of life and plants and organic materials in your system, you probably find that you don't need to do these culturing practices so much anymore, because it's just happening naturally. You won't, you'll find your need for compost will be less. You won't need to use as much foliar sprays because all those organic acid processes are happening in the soil, and you'll have like good nitrogen fixation happening from rhizobia, and your plant health will be better, so you don't need to use as many products for that as well. Your leaves will be colonized because they're germinating, in an environment that's rich in beneficial organisms. The plants will be colonized with beneficial organisms. You'll have less problems all around. So it's kind of like you're working towards creating a system, as I said before, that's going to do this for you. But these are little shortcuts to help you move your system to that point. Using grazing animals is great because they really incubate and spread microbes in their guts, and then the manure, as they spread it out, and using manure as an inoculant in your compost and even in your gardens is a great thing. And then you can actually, saving your own seeds is a great thing to do too, because what they've found now is that the seeds of plants actually, the plants will actually put the microbes into the seeds that are going to be beneficial for them when they germinate. So if you're growing healthy plants in a healthy environment and you save the seeds from that, it's already going to have a strong, healthy seed microbiome. And when that seeds germinate, it's naturally going to already have those microbes in situ, and they will then be in the soil area the environment that you introduce those seeds to. So where you can, saving seeds is great, or even vegetative propagation is a great thing too from plants around the place. Whether you're using like garlic or tubers, all of those things are colonized with microbes, and it's certainly much better than bringing in garlic from China to plant that's been possibly radiated or something like that and is sterile. And when I'm making potting mix, I often use garden soil as at least 50% of the potting mix. It's cheaper, it's got a whole lot of microorganisms in it. And then I will add something like compost to that as well, which has got all those benefits, and it just grows excellent seedlings. People worry about using existing soil because it might harbor diseases, but when you get your soil to that level of health and the expression of the organisms that are in there, you just tend not to have those problems. And this is not for commercial nursery stock, it's just for growing seedlings for your own situation, so like a veggie garden or an orchard or whatever. And yeah, it just works really, really well. using compost and inoculants at planting, as we talked about, is really important. And then if you are making foliar sprays or liquid, putting some liquid fertilizer out through your fertigation unit or just drenching your soil, always include compost extract if you can, because it's full of biostimulants, it's full of really beneficial dormant organisms, and if you're adding foods to that compost extract as well as your nutritional applications, those organisms in the compost extract will germinate, and they will colonize surfaces of leaves and roots and all parts of the sort of the growing plant sort of environment, and just again make for really healthy a healthy plant microbiome. So yeah, you kind of want to, again, in summation, we haven't really bred specific organisms that we know exactly what we're culturing, but we're through all of these cultures, we're growing a whole lot of really beneficial microbes, and we can use them in a range of situations, not just as an inoculant, but also because of how they can improve, they create these other substances that we can use, like organic acids, and also the biochemical component of them when we use them out in the environment that stimulates microbe activity. So that's the presentation, folks. I didn't see any questions. Kate didn't stop me, so now's the time.
That was a really fantastic, Mark. And actually, who knew when you were going into farming that you would also have to be a biochemist?
Yeah, but you don't need to have specialist skills. That's the good news, Kate. It's quite simple technology that, it's quite general principles that we learn and very easy to apply and cheap.
Yeah. Well, I'm sure people have got questions, but while people are sort of thinking about what they might want to ask, I've got a couple of things that I wrote down as we were going along.
Yep.
And one of those was right back at your early start, you were talking about compost and compost maturity.
Yeah.
So what sort of time frame are you talking about for a compost to mature?
It all depends on the compost method you use. So if you use say the turning method, the sort of soil food web type model where you're turning it a lot at the very beginning and it's going through a very hot phase, and every time you turn it, you reintroduce any materials that haven't already been eaten get put into the middle, and it's a quicker process. So you can actually make a compost that way, within a couple of months, sometimes even up to a month, and it can be really quite a good compost to use. However, it would still benefit from sitting for longer and the introduction of worms. And when you go into something like a Johnson Sioux, which you're not turning, it preferably six months to a year before it's gone through all those processes and worms have gone through it. And by then it's a culture, not just a compost culture, but a vermiculture as well. And it's just colonized by fungi all through it. So yeah, it depends.
And all that time it needs to be left alone pretty much after the thermofit.
Yeah,
so you really have to have a spot that you've turned it into and you're happy to leave it there.
Yeah, and you have to keep an eye on the moisture, and you have to make sure that it's out of direct sunlight maybe as well in the hot summer. So yeah, those basics, again, they're all covered in the composting doc, which we'll send out. But yeah, really sort of, depending on the method, it can be anywhere from a month to a year of what qualifies as maturity.
And how would you rate, how would you describe the difference between vermicast? Let's say you wanted to use extracts.
Yep.
What would the main differences between vermicast extract and compost extract? Interchangeable or?
Most of the vermicast extracts we buy commercially are actually composted first and then vermiculture added, and that's the best way to do it because it's just a broader range of microbes, and you also kill some pathogens in the heat phase. I mean, not many pathogens survive once they've gone through a worm's guts, but it really does help to put your compost through a bit of a thermophilic phase as well, and yeah, just greater diversity and that sort of thing. So I think vermicasts by themselves are great, but again, you have to maintain an aerobic environment. A lot of worm farms are not as aerobic as they need to be. They're too wet, and they get a bit saturated and rotty. So I think they can, either way it's great. And like a really simple worm farm would be just like you put down like some weed mat or something that perforated on the ground, and then you just layer a foot of like straw and manure and green stuff, and then you cover it with some hessian or weed mat again, and introduce worms to it, and you leave it for 6 months to a year, and it just comes out beautifully. It's so simple. So yeah, either way, whatever's going to work for your system. But if you've got the capacity to make compost and then I love to do that, make compost and then vermicast, put worms in once it cools down.
Yep. Well, it looks like we've got lots of people asking questions now, so we can move on to those.
Yep.
So Steve asks, how important is water quality for compost extracts and slurries? Thinking bicarbonates more so than chlorine.
Yeah, less so than for foliar sprays, because the reason why you need to deal with your bicarbonates is because you're adding sulfates and salts, which then react with the bicarbonates and become plant unavailable again. Whereas in the compost extract, you've got really stable organic molecules that are you're less sort of focused on the nutrients, although there are nutrients in there. You're more focused on the biochemicals and the dormant microbes, and the bicarbonates don't have much effect on those. So you can use just about any water with your compost extract. But I mean, rainwater is always great if you've got it.
And we have from Colin, what is the use of aerobic compost tea versus anaerobic mix? I think you kind of answered that, didn't you?
Yeah, I think that the place for anaerobic like brews is more in the chelating organic acid, complexing and making nutrients available. So more using them for putting nutrients in the right state, whereas an aerobic mix is more about a biostimulant and an inoculant. So when you're doing compost extracts, it's more for the microbes and the biostimulant sort of compounds in there. Whereas when I make anaerobic brews, I mostly use them with nutrients to get nutrients into the right state for foliar sprays and soil drenches.
Okay. And Jason asks, is it important where you source your milk from?
Not really. You can use milk powder, you can use whey, you can use milk, you can use skim milk, whatever.
The only thing is about the milk powder, I remember when we were making our own brew up, Mark, the sediment settled in.
Yeah. I mean, if you can use whey, it's even better because there's less sediment in it. But yeah, obviously sediment is part of using milk. And you have to have like a tap above the bottom of your tank so you can use your, decant your biofertilizer without getting the milk solids into the mix.
Yeah. So what's the application rate for broadacre sprays? Peter asks.
You're talking, it's hard to say whether he's talking about foliars or extracts there, but standard extracts, sort of like 1 kilo of compost or a litre of compost, will cover a hectare for an extract spray. And you're looking at things like, when you're adding fish hydrolysate or fulvic acid, it's sort of in the vicinity of 1 to 2 litres, depending on whether you're broadacre or horticulture. You can sort of up the rates a bit in smaller situations. And then in terms of your nutrients like sulfates, you just have to know what your, I put a few on there, and maybe if we send out recipes, we can include rates that are used as foliar sprays, but you just have to do your research and know what your rates are, which most farmers are well familiar with when it comes to foliar sprays. Understanding that you can err on the low side because these are really, really readily plant available. And so, better to put on more frequently at lower rates, highly plant available, than putting big hits on at one time.
Yeah.
Yeah.
Katrina's asked, how long is too long to store your biofertilizer? She says mine is being used up and is 1 year old.
Yep, at least a year, I'd say, depending, maybe, up to 2 years, maybe. You just have to check it and just see how it goes. Provided it's cool, not too hot, stored under cover, and it should be right. But the other thing is, you can always, if you've been storing it for a year and you're a bit worried, you can just add more milk and molasses to it, and it'll get going again, and the microbes will come alive and do what they need to do, and then they'll go dormant again, and it'll probably last for another year or two after that.
Yeah, and actually someone did write in, because we changed the time.
Yeah.
They asked for a question to be asked, so that kind of segues into that. How long will live microbial extracts or teas remain viable without aeration or stirring? And is there a way to put them to sleep, as some commercial companies claim?
Yeah, I mean, the teas, when you talk about tea, normally it's used in the sense of aerobic. So that you get compost and you put it in a brewer with a bubbler and you add foods, and it has to have a certain level of oxygen for those microbes to survive. You cannot store that. You have to apply it straight away after you've made it, within hours. Whereas an extract, because you're dealing with dormant organisms like spores and cysts, you can put them in a liquid and they do last a lot longer, but you don't want to add foods to that and then store it. As soon as you add foods like milk or molasses or fish or fulvic acid or whatever, you have to spray it out. And as to how long a good compost extract from a mature compost will last, it's hard to say. I prefer to use them fresh, but I haven't really got the exact information on how long they store for. But I've certainly stored them for a few weeks and even months at times, and then used them, and they don't seem to have any bad growth or bad smells or anything like that.
And teas are slightly different anyway, aren't they? Because they're brewed as opposed to extracts. Yeah,
they're specifically dealing with very aerobic organisms, and that you do have to know what you're doing with a tea, in terms of you have to be a lot like with the extracts and with the biofertilizers. They're very hardy cultures. I mean, I know you made some biofertilizer and you left the lid off, Kate, and it's still good, isn't it?
Yeah.
And the extracts are the same. They're very
Don't do that at home, folks. Put the lid back on.
It's still good, isn't it?
It is still good. Still smells sweet. Yeah.
Yeah. So they're very hardy things, and extracts are a bit the same. They're not quite as hardy as a biofertilizer, but they're, if they're in the dormant form and they're stored in cool conditions, they should last for a while like that. And also the other thing is, because they're full of the biochemicals, those biochemicals are fairly stable. They will over time slowly sort of, there will be reactions and things that happen that they get broken down, but you're not just applying dormant organisms, but you're also applying these biochemicals, which is an important part of extract. So you can store them in water for a little while before you apply them. But I mean, I always make my brews as fresh as I can. I love to get, because there are some living organisms even in a very mature compost anyway. There's always going to be some life there.
You do almost, well, you need a kitchen outside. You need your
You
farm kitchen
do.
with all
And
that stuff there.
nice and organized. Okay, makes it a bit
That's
easier.
right.
Yeah.
So Phil asks, hi Mark, how does one extract rumen contents? And thanks also for the presentation.
Well, it's not for the faint-hearted, but anyone who's dealing with livestock, and if you kill an animal and you basically just have to get the stomach and cut it open and you pour the rumen contents into the barrel, it's that simple.
Stinky.
Yeah, it's a stinky job, but there's lots of cultures you can go with if you don't want to do that, which are quite easy. And the other thing is, once you have a biofertilizer going, you just use that as a starter culture next time as well. So really, you could just start one with kefir and yeast, and that would do the job. Or get some BAM and use some of that, or effective microorganisms, and then apply that. And once you've got a good culture going, it's like a sourdough or a kefir culture. It's very easy to just use it as a starter and keep feeding it and keep it going.
Yeah. Jock asks, are meat hydrolysates different to fish, and will adding sea salt or kelp help?
Yeah, I mean, they've all got different properties. The fish might have, a few more minerals from the ocean in it, but yeah, essentially it's a protein thing. So whether the bones are in it as well, you might have a calcium and magnesium element and phosphorus. So yeah, I mean, but essentially, hydrolysis is you're dealing with proteins. So, or hydrolysates, sorry. So yeah, they're great either way, fish, meat, whatever.
It's just a comment, not a question. Top work, Mark. So informative, very precise, makes sense holistically of many different complex interactions. Can't wait to spread the good word. Thanks, Peter.
Thanks, Peter.
Jock also asks, molasses is hard to get. Is sugar an adequate substitute, and why?
Okay, I haven't found molasses hard to get myself. I find it readily available from stock feed places and rural sort of agricultural suppliers. But yes, you can use sugar. Yes. Molasses has more minerals in it as well, and a few B vitamins and things. It's generally better for the culture, but sugar does work.
Bob thanks you very much for his presentation. But he also says, by the way, I use my urine as compost activator. Will contain my multivitamin supplements.
Yeah, urine's pretty sterile, but it's full of urea. So it's a great protein source to add to your compost and your fruit trees and all that sort of stuff.
Thanks very much for that, Bob.
That's really good.
That's really utilising everything.
Yeah.
Well, I think that looks like we've come to the end of the questions there, which is great.
Yeah.
Just seeing on my little session. Oh, I did have a couple of questions / comments. In a grazing scenario, and I know this from my own experience, I sprayed out my own BAM mix after I'd grazed, and then you said, well, I should have done it before.
Yep.
Can you just explain to the audience why that?
I guess when you graze your field, you're getting rid of a lot of the foliage, so a foliar spray is going to be less effective. And then also there's naturally going to be a lot of root exudation and activity that comes with the graze. So when you apply it before the graze, you get better intake into the leaves, but also it sort of stimulates the plants and makes them more nutritious when the stock do come to graze them. So you're kind of like giving the stock the benefit of the foliar application as well. That's the main reason, I think.
And one last thing before we move on, I think when if you haven't done any sap testing, were you talking about using a general kind of mineral supplement or whatever? You can do little sort of trial strip tests, can't you? I mean, you can make up a little brew with something in it, spray it on a small patch and see what sort of reaction you have, and do a bricks test or something to see if it's had any impact.
Yeah, you can, Kate. There are a few nuances to it, and I find most people don't. So it's not something that I would sort of espouse, because I want this to be really accessible and to work for people. One thing I will say is that when you're doing foliar sprays, really to get the nutrients that you need in the adequate quantities into the plants, you're primarily dealing with trace elements. So, because they're a foliar spray, you're applying them at such low rates that even if you don't have an issue with that particular element, you tend to be okay. You do have to watch some of them, like boron. because, you know, like that, any element, if you put too much of it, can burn the plant, and if it's got a lot of it in there. But if you err on the side of, like, when you do general purpose foliar sprays, you generally use lower rates of nutrients than if you're just doing a single application of something like iron or manganese or something like that. So you would do a broad range, and it's in low rates, and you apply it, and it's just a tonic of nutrients that gets them into the leaves, and it really, you know, they can be really beneficial having a, you know, just a general purpose trace element foliar application. But there are specific nutrients that are associated with certain processes that might be helpful. So like if you're having particular issues with photosynthesis, or you know, you've got too many nitrates in your plants, like protein conversions looking like a problem, and you've got lots of sap-sucking insects appearing on the scene and stuff like that, there are certain nutrients, you know, you can target. It's just probably a bit beyond the scope of this presentation to go into that.
Yeah, sure. Sure. Okay, well, I think that's probably come to the end of things. So, and very timely given the rising cost of ferts. But I also know that many people, including myself, like to learn by doing as well. So as Mark alluded to, we do also hope to do at least one hands-on workshop on this same topic later in the year. So keep an eye out for that one. So thanks once again, Mark, for a brilliant presentation. We'll let you go to get on with the rest of your day. For our audience, before you sign out, I'd really like you to complete our survey. Just find the right one. That one there. It's important for us so that we can get good feedback and keep providing the content that you want. It'll only take you a couple of minutes, and it's really super helpful. And I hope to see you in our next event. For your information, we've got Dr. Christine Jones on a similar theme, talking about on the fertilizer theme. She'll be joining us on the 4th of July, talking about the impacts of adding inorganic nitrogen and phosphorus to our soils and how we can wean ourselves off them. So I do hope that we'll see you there. All right, so that's it for now, and thanks everyone, and hope to see you next time. Thanks, Mark, again.
Thanks, Kate. See ya.